In 8.2, we will talk about the light-dependent reactions, which is the first part of photosynthesis. Before talking about that, we want to talk a little bit about light and the kind of energy that our pigment will absorb. So we have the electromagnetic spectrum, which is all the range of possible frequency of electromagnetic radiations. As you can see here, from a really high energy gamma rays to X-ray to UV light, which is what you sort of hear a lot about, that can damage your DNA and cause mutations within your DNA and cancers. Up to visible light, to these visible light, which is the one that we see, from red to all the way to violet. And then we have our radio wave, which would transmit your radio. And before that, you also have like infrared, which is usually used for night visions. So all of these are possible wavelengths of light. However, we only concern about the visible wavelength of light for our photosynthetic pigment. So within this visible light, so this is a higher end, whereas your UV and X-ray exist, and this is the lower end where you have infrared and then radio wave. You have your red wavelength, orange, yellow, green, blue, indigo, and violet. So this is basically the rainbow. So you have a much longer wavelength, starting at red, around 700 nanometer, all the way down to the 400 nanometer, which is the shorter wavelength. That's your violet. The plant photosynthetic pigment can actually absorb most of these wavelengths, and of course, you probably can already think of, they absorb the lowest at the green wavelength, is because they actually appear green, as you can see in a little bit. They reflect and transmit that green colors. That's what we see. It's important to see these wavelengths, because the longer the wavelength, the less energy they have, and the shorter the wavelength, the more energy they would have. So if a wavelength has too low of energy, they will not be able to excite the electron that would be used to make ATP. And then if it is too high, then it would damage or bleach our actual photosynthetic pigments, and that would not be a good thing. So the pigments that we have been talking about that are responsible for light absorption is going to be divided into two major classes. We have our chlorophylls and our carotenoid. And so our chlorophylls, we have chlorophyll A, B, C, and D, but you will hear the most about chlorophyll A and chlorophyll B in photosynthesis, because those are the main photosynthetic pigments inside the leaves. So they are essentially identical, except this part in the box right here. So you see that they are essentially the same exact molecules. And then we have our carotenoid, which are essentially what gives a reddish color, orangey color in the leaves. So these are mainly the pigments that you see in the fall. So you found them in tomato, you found them in corn, you found them in orange peel and carrot. So the main one that we will see will be beta carotene. They are basically there in order to absorb the excess energy and then safely dissipate that as heat, because plant cannot absorb all of the wavelength, and if they have too much energy and they cannot absorb that, the leaves will be damaged. So beta carotene is there in order to help out with the excess energy and make sure that it's not there to damage the plant, and they would actually will be able to perform photosynthesis safely away from the heat, essentially. So this is the spectrum that you saw before, from around 400 to roughly about 700. So this is red, this is violet. These are the spectrum. Here you'll also have the absorption ability of these different photosynthetic pigment, chlorophyll A, chlorophyll B, and beta carotene. And as you can see, chlorophyll A, actually A and B, they mainly absorb in the bluish, so this is a solid line and the bigger dashed line, so bluish and reddish region, and they don't really absorb anything in the green area. So they will absorb that, and that color will just basically become invisible to the eye. Because they are absorbed, and the color that are actually reflected or transmitted, they will be what you see. So because they're reflecting green and transmitting that green color, they will appear as green. So beta carotene, they reflect these three colors, so they generally appear as reddish, orange-ish color. That's the leaves color when you see in the fall when chlorophyll sort of breaks down. So most of the leaves, they have a mixture of all of these three or even more photosynthetic pigment. They don't just have one particular kind. But one thing to keep in mind is that you can see here in this absorption spectra is that each pigment has their own and unique absorbance spectrums, and they prefer or they only absorb a certain kind of wavelength. They don't absorb everything. Thus, they give that a color appearance for the color that has been reflected and transmitted out back to your eye. Okay, so let's talk about a little bit. Start on our light-dependent reactions. So you need to be familiar with the concept of a photosystem, which is a groups of proteins, which has chlorophyll and other pigment as well, and they are used in our light-dependent reactions in order to absorb sunlight energy and then convert them into chemical energy. That is using a photosystem to perform the light-dependent reaction. So you need that input of sunlight in order to make it happen. So you have our photosystem 2 and then photosystem 1. So photosystem 1 is actually discovered first, however it's come later on in the pathway. So 2 usually is put first and then 1 is putting after, but their naming is sort of reverse. And these, you can find them in the thylakoid membrane. So the general structure for this, you will have the light harvesting complex, which are these green parts right here with the pigment molecules. You can see that in 2, you can see that in photosystem 1 as well. These are responsible for absorbing light. So light will come in as photon, which is essentially package of light. Lights act as a wave, and they act as a sort of a particle-like properties as well. So lights come in as a photon. They will be absorbed by these pigment molecules, and then they will transfer that energy to the reaction center, where they will have special pairs of chlorophylls that will be responsible for excite an electron to pass that to the primary electron acceptors. So that is the reaction center right here. So within photosystem 2, our reaction center will have the P680, and within our photosystem 1, we will have the P700. You see that more in a little bit. As you can see here, this is a chlorophyll A pair. So these are special chlorophylls that are that acquire enough energy to excite that electron. So we will go through each steps of the light-dependent reaction, starting with the photosystem 2, then we'll have an electron transport chain, and then we have our photosystem 1, then we have the rest of the electron transport chain. And we'll see in action how do they absorb light and actually produce ATP and NADPH. So this is step one. Step one is the light absorption within our PS2. This is our photosystem 2 with the reaction center P60. So you have our pigment absorb light in terms of photon and pass that energy to the reaction center. So you see here light coming in, transfer the energy to these two chlorophyll special chlorophyll pigments, and they will use that energy condense here and excite an electron that you can see right here, and that electron actually comes from the splitting of water. So you split water into oxygens and hydrogens, and you will get 2 electrons, and you use the energy here to excite that electron and then pass that electron to the primary electron acceptor. So the job of this step is essentially excite this electron to a higher energy state. So make that electron store energy within that electron. And then for the next step, that electron will be passed down from a bunch of protein, like what you saw in the electron transport chain in cellular respiration. This is the same process. You have high energy electron that will be passed down into this electron transport chain. They will lose electron as it go down this pathway. However, that energy is not being lost. They will be used to import that hydrogen ions from the stroma into the lumen of the thylakoid. So you see that import of hydrogen ion, and this H will actually contribute to that hydrogen ion gradient as well. And as you remember, a chemical gradient will have potential energy that will be able to go down an ATP synthase and actually make ATP. So this is the same process where you import hydrogen ion, making that gradient, and that gradient will diffuse down across that ATP synthase and make ATP through the process that we talked about before called chemiosmosis. So that's the second step, where you have our electron transport chain and ATP synthase. Then in the third part, at this point, our electron has lost energy. They go down in their excitement state. So what they do is, instead of just putting this electron somewhere else, they actually use another system called photosystem 2 with the reaction center P700 to actually excite this electron again. So our photosystem 2 received the electron from the splitting of water, while the photosystem 1 received the electron from the electron transport chain, which essentially originate from the photosystem 2. So same thing happened as what would happen in photosystem 2. We have a light energy here in terms of photon passed down into a bunch of photosynthetic pigments here, pass down to that reaction center, excite that electron again, and pass that to an electron acceptor. As this excited, it will go on to the last steps of our light-dependent reactions. So as you can see here, this is a replenishment of that electron coming up from here, which are arriving by the electron transport chain. So here you have the last steps of our light-dependent reactions, where you will use that newly excited electron, and they will go down on the rest of the electron transport chain until they reach the NADP plus reductase, and this is an enzyme that will catalyze the reactions of NADPH and with an additional hydrogen ion to convert that into NADPH. And this NADPH and the ATP that you made before in the electron transport chain and chemiosmosis, you will use that for making glucose molecules in the light independent reactions, or the Calvin cycle. And this is the overviews of the light independent reactions. We didn't really see this part in this steps, but as you can see, we split water, getting that electron. That electron is excited by photosystem 2, passed down an electron transport chain, making that hydrogen gradient in order to pass down to ATP synthase to make ATP. This electron is now losing a lot of its energy and go down to its into energy state. They will be passed down to photosystem 1 to be excited again, and then that electron will be passed down to an NADP plus reductase, where they will facilitate the reactions of NADP plus and an hydrogen ion to make NADPH. NADPH and ATP, these are energy molecules. They have successfully take light energy and convert them into bonds of an organic compound in forms of chemical energy. They will be used to make sugar precursors, or essentially to form glucose down the line in the Calvin cycle. We will talk about that in 8.3.